Hey everyone, Dr. Chem here, and welcome to today's Explainer. Today, we're going to be stepping into the shoes of molecular detectives. Seriously, just picture yourself walking into this cutting-edge, futuristic analytical chemistry lab. The air is literally humming with the energy of superconducting magnets, and all around you, these glowing animated lines are tracing the invisible architecture of organic molecules. You can see the carbon and hydrogen atoms, all intricately connected within these super complex compounds. It's amazing. And right at the center of all this is what we're decoding today. One of the absolutely most powerful tools we have for modern structural elucidation. We're talking about DEPT NMR spectroscopy. Okay, let's dive right into this. Just look at this absolute beast of an instrument. This is our primary technological weapon, the Bruker 500 Ultra-Shield Superconducting NMR Spectrometer. Inside that massive cylindrical housing, it's a super-cooled environment generating incredibly strong pulsing magnetic fields. Now, this machine doesn't just, look at a molecule. It actually interacts with the quantum spin of its atoms. It surrounds them with these invisible lines of magnetic force. But to really get why this specific machine's deep cap capabilities are such a massive game changer, we first need to talk about the huge blind spot it was actually designed to fix. Think of your standard broadband decoupled carbon-13 NMR spectrum like a sort of incomplete map. Don't get me wrong, it's fantastic at telling us exactly how many different carbon environments exist in a molecule. You get this nice, clean, vertical peak for every single unique carbon atom. But here's the catch. Our central mystery, or the villain of our story, if you will. That standard spectrum completely hides how many hydrogens are actually attached to each of those carbons. I mean, looking at a standard peak, we literally have zero clue if we're dealing with a methane, which is a CH group, a methylene CH2 group, a methyl CH3 group, or even a quaternary carbon that has no attached hydrogens at all. They all just look like identical lines on the page. So, to solve this mystery, we need a major technological upgrade. Enter DEPT. That stands for Distortionless Enhancement by Polarization Transfer. Okay, I know it sounds like a massive mouthful, but the actual concept, it is incredibly elegant. DEPT is essentially a specialized pulse sequence that differentiates carbon atoms based strictly on their attached hydrogens. It takes that flat, uninformative carbon map and injects this crucial extra dimension of data. It tells us exactly who is partnered with who down at the atomic level. To really appreciate how wild this is, we've got to look at the quantum mechanics going on under the hood. The physics of polarization transfer is kind of like this microscopic energy dance. See, carbon-13 nuclei are notoriously lazy. Their resonance is roughly 6,000 times weaker than that of a proton, or hydrogen nucleus. So what DEPT does is it exploits the heteronuclear J-coupling, basically the magnetic connection between the carbon and its attached hydrogens. Imagine we're firing highly precise radio frequency pulses that align the spins of those sensitive, abundant hydrogen atoms. And then we literally transfer that excited magnetization, like rotating energy vectors, straight over to the weaker carbon nuclei. This massively enhances the carbon signal. And the best part? Because this transfer requires a hydrogen atom to act as the donor, any carbon without a hydrogen, those quaternary carbons, are completely suppressed. They just vanish from the spectrum entirely. By tweaking the tip angle of the final radio frequency pulse in the sequence, the spectrometer basically acts like a camera, swapping out different filters. It gives us 3 distinct lenses to view our molecule. We do this through specific rotation multipliers applied to that final pulse, creating this elegant dance where magnetization vectors rotate, evolve, and transfer polarization. A 45-degree rotation gives us the DEPT 45 experiment. A 90-degree pulse gives us DEPT 90, and you guessed it, a 135-degree pulse gives us DEPT 135. Each of these specific angles interacts uniquely with the complex quantum spin dynamics of those CH, CH2, and CH3 groups. So the crucial point is really this master cheat sheet, which shows exactly how these filters work in practice. Let's walk through it together. In DEEP 45, every single carbon with an attached proton, so your CH, CH2, and CH3, shows up as a positive peak pointing upwards. Quaternary carbons, entirely absent. Then we move to DEEP 90, and the filter gets a whole lot tighter. Here, CH2 and CH3 signals vanish entirely, leaving only the CH, the methane carbons, pointing up. But the true star of the show is DEEP 135. In this one, CH and CH3 carbons point up as positive signals, but the CH2 methylene carbons actually invert. They point downward, forming a negative signal right below the baseline. And again, across all 3 of these experiments, quaternary carbons remain totally invisible. I really want to pause on this for a second because it's such a beautifully visual piece of science. In DEEP 135, that inversion of the CH2 carbons makes them instantly recognizable. You don't need to sit there doing complex math. You just look straight at the baseline. If the peak is plunging downward like a stalactite, boom, you have a CH2 group. If it's pointing up like a stalagmite, it's a CH or CH3, which you can then easily separate just by cross-referencing with your DEPT 90 data. It's so clean. Now, before DEPT came along and totally revolutionized the laboratory, chemists were stuck using this much older technique called off-resonance decoupling. In this method, the decoupler would irradiate the sample at a slightly off-center frequency. This avoided complete decoupling, which allowed the carbon to split its signal based on the number of attached hydrogens, following the classic N+1 rule. So under that old system, a CH3 would show up as a quartet, a CH2 as a triplet, a CH as a doublet, and a quaternary as a singlet. Now, what's really interesting about this slide is how quickly that old method turns into pure, unadulterated chaos. Just look at this off-resonance decoupled spectrum for 1-propanol. It's a relatively simple molecule, right? But the messy peaks spread out and they constantly overlap with each other. If this is what happens with a simple 3-carbon molecule, just imagine trying to decipher an off-resonance spectrum for some complex pharmaceutical drug with 40 carbons. It's a total visual nightmare. This perfectly illustrates exactly why the clean, single-line precision of DEPT was so desperately needed in the field. All right, let's plunge into a real-world puzzle to see our molecular detective skills in action. Say we are handed a mystery sample with the chemical formula C5H9Br. From our initial chemical analysis, we know for a fact it has a double bond, but we don't know exactly how the carbon chain is actually arranged. Based on standard proton and broadband carbon NMR, we're totally stuck guessing between 3 perfectly plausible structural candidates, and they only vary by where a single methyl group is placed on that double bond. So standard methods have totally failed us here. The tension is high, right? We need a definitive answer. So our lab technician runs the ultimate lie detector test, a DEPT 135 experiment on our sample. Remember, we're looking for those incredibly specific structural fingerprints. The results come back, and they are striking. The spectrum clearly shows exactly 3 negative signals plunging straight below the baseline. And we know immediately what those negative peaks mean, don't we? Because DEPT 135 showed exactly 3 negative peaks. Our mystery structure absolutely must contain exactly 3 CH2 or methylene groups. Looking back at our 3 plausible suspects from earlier, 2 of those molecular structures only have 2 CH2 groups. Instantly, they're eliminated. The final verdict is crystal clear. The only structure that perfectly matches our DEPT data is the one containing 3 separate CH2 units. Mystery solved, definitively and rapidly, just like that. Let's move to another case file and see how this builds, proving that our structural logic is universally applicable. This time, we're looking at an enigma with the formula C3H6O2. If we compare the proton decoupled baseline with the off-resonance data for this molecule, we see 3 distinct sharp lines. Those are our 3 carbons. When we look at the off-resonance spectrum, we can actually track these signals as they split. The signal furthest to the left stays a single line, a singlet. That means it's a quaternary carbon with absolutely no attached hydrogens, very likely our carbonyl carbon. The other 2 signals split into distinct quartets. Using that N+1 rule we talked about, a quartet means N equals 3 hydrogens. Therefore, those 2 signals correspond to CH3 methyl groups. By cross-referencing these visual cues, whether we're using DEPT's incredibly clean lines or tracking these messy splits, we literally reconstruct the atomic reality of the molecule. So, what does this all mean? Why does all this matter? Why go to such crazy lengths to map out this invisible architecture? Well, because in modern laboratories, knowing the exact connectivity of carbon atoms isn't just some abstract academic exercise. This rapid, distortionless structural elucidation is the very engine driving modern innovation. It allows chemists to verify they've successfully synthesized a new life-saving pharmaceutical drug without any dangerous isomers slipping in. It's absolutely crucial for quality control when designing next-generation synthetic polymers, and forensic labs rely heavily on it to precisely identify unknown illicit compounds. DEPT literally turns guesswork into absolute structural certainty. As we envision a future where high-powered magnetic fields and rotating atomic resonance continue to push the absolute boundaries of chemical research, you now possess the analytical logic to look right past the surface of a spectrum. You understand how manipulating the spin of a single hydrogen atom can light up the entire hidden skeleton of a carbon chain. You're officially empowered with a brand new set of highly advanced detective skills. So the only question left to ask is, what invisible molecular mystery will you decode next?